Hello, everyone, and welcome to our webinar. We are going to have a very interesting topic today where we discuss what might be the sort of origins of Parkinson's disease. Where does it really begin, or where does it start? And we have a great set of panelists who are going to be talking. First of all, I should introduce myself maybe. I'm going to be the moderator, but I will also be chatting a little bit. My name is Patrick Brundin, and I'm in Grand Rapids, Michigan, where I direct the Center for Neurodegenerative Science, and I have a very long-standing interest in Parkinson's disease. And in this context, I'm actually interested also very much in where the disease starts. But the first of our panelists I want to introduce you to is Dr. Filip Scheperjans, who is a clinical neurologist in Helsinki. Can you say something, Filip?
Hello, welcome everybody.
Welcome, Filip. And Filip is very much an expert on gut microbiome, which is a super hot topic. Our next panelist is Dr. Jon Stamford, who is doubling up both as a neuroscientist and a person affected by Parkinson's disease.
Hello
Jon,
there. Hello, good to hear from you. Hopefully, as said, I shall interject from time to time. I'm more a person with Parkinson's disease than a neuroscientist, I think, these days.
And finally, in my screen, furthest to the right, I have Dr. Viviane LaBrie. Dr. Viviane LaBrie is in our center here in Grand Rapids, Michigan. So although she's in a different screen, she's only about 40 feet away in an office close to me. And Viviane is an expert on origins of Parkinson's disease and epigenetics, which I will let her explain a little bit later what that is. And can you say hi, Viviane?
Hello, everybody. As Patrick mentioned, I'm an expert on epigenetics, and I also try to understand the mechanisms of origins of Parkinson's disease. Welcome.
All right. So also to everybody who's listening in, we want to remind you have the possibility to pose questions as we are speaking, and you can pose them by using the chat function. And I think you can use something called all attendees. At least I think that's what it will be. And if you just type in a question there, it should reach me and the other people on the panel. And it should also reach the Cure Parkinson's Trust, who are represented by Helen Matthews and Julia and Eros, who are all in London. But you can't see them. They can see you and hear us, I should say. All right, let's kick off with a discussion about where Parkinson's disease might start. And let me just set the stage by saying that when I started as a doctoral student looking at Parkinson's disease in the 1980s, we were really only interested in death of dopamine neurons that are in what's called the midbrain, so somewhere right here in the middle of the head. And we knew that those cells govern motor function, certain aspects of motor function, and when they're lost and the dopamine that they produce is absent, it becomes difficult to move. So there's the stiffness, there's the slowness of movement, and the tremor. And we were totally obsessed with these dopamine neurons, looking at various ways of finding out why they die and what could be the triggers that cause them to die. And there was a big hoopla in 1983 when a group of heroin addicts in California made what they thought was a synthetic heroin called a meperidine analog, which would have been the synthetic heroin. But accidentally, there was a contaminant, which turned out to be a chemical called MPTP. Those very unfortunate heroin addicts, at least 7 of those who injected this drug, developed acute Parkinsonism. So they showed all the motor symptoms of Parkinson's disease within 1 or 2 weeks. This was perhaps the first time we saw what could cause the death of dopamine neurons in humans. And we became very interested in this. And, the idea was that perhaps it was a molecule that looked like MPTP, like a pesticide or some environmental toxin like that. And the whole field was then focused on trying to understand how this environmental pollutant could attack those cells in the brain. So what's happened since then is that we've gradually realized that Parkinson's is much more complex. And I think virtually everyone listening to this webinar knows that it's not just about motor symptoms. Instead, there are several non-motor symptoms and signs of Parkinson's. And we've learned that these frequently precede the disease diagnosis. So before anyone ever goes to the doctor complaining about the tremor or the stiffness, they will have exhibited, for example, disturbances of sleep, constipation, and loss of sense of smell. And depression is actually also a common feature of this, which we call the prodromal phase of the disease, so before the formal diagnosis. And with the realization that that prodrome can go on for 5 to maybe 20 years, we have sort of, as a research community, decided that we need to understand this prodrome, and maybe the enigma surrounding what triggers the disease, the answer to that enigma is in the prodrome, because those parts of the nervous system that are involved in the prodrome are likely to be the parts where the disease actually starts. So that was a long introduction, but it's going to tee off, I think, the first topic, which is, as you know, we have an article that you can read that is a bit coupled to this, How Does Parkinson's Disease Begin? By Per Borghammer. And in that article, he discusses a lot about the gut and constipation being a very common feature of the prodrome. So I'd like Philip to talk a little bit about the gut in Parkinson's disease, first of all, and constipation, and maybe then tell us a little bit about his work on the bacteria that we normally have in the gut, the microbiome.
Sure. Thank you very much for the introduction, Patrick. Yeah, I think what we have realized is that constipation is actually probably one of the most common non-motor symptoms. So it has actually turned out that when you measure objectively transit time, for example, in Parkinson's subjects, then about 80% of patients will show signs of constipation, even though some of them don't actually suffer any symptoms, so they don't realize that they have a slowing of transit. And for me personally, the most compelling finding is that those patients that complain about constipation, at least half of them would describe that this has began before the onset of motor symptoms. And we have quite convincing data that there may be even several years, some studies say up to 20 years delay after the onset of constipation and the motor symptoms of Parkinson's disease. So I think epidemiologically, it is quite compelling. And also there are studies that suggest that an idiopathic constipation, so that you cannot explain with any other reasons, that it increases your risk of Parkinson's disease about 2 to 4-fold. So it's, I don't know whether you would call it a risk factor or if we consider it an early symptom, but it at least is associated with an increased risk of Parkinson's disease. And actually, in our own work, we have also looked at a syndrome that's called Irritable Bowel syndrome. And at least we have found that it turned out that actually many of the patients that report constipation, when you give them a questionnaire that actually assesses irritable bowel syndrome, then they turn out to actually fulfill these criteria, quite a majority of them. And so it may actually be that a significant fraction of these constipated patients is not only suffering from constipation, but from irritable bowel syndrome. And I think that's pretty interesting, because irritable bowel syndrome is the archetype of a so-called gut-brain axis disorder. So it involves disturbances of gut function that you cannot trace with classical histological stainings, for example, at least not in clinical routine. There's quite strong evidence that microbiome changes play a role in irritable bowel syndrome, but it also involves the central processing of sensory input from the gut. So there's also a central component to irritable bowel syndrome. And there is quite good evidence that by certain manipulations of let's say, diet and so on, you can influence these symptoms. And so this may actually be one of the less recognized symptoms in Parkinson's disease. So in many patients probably not suffer from simple constipation. But then it comes up with, the question is obviously, what's the hen and what's the egg? So is the constipation let's say, a trigger for Parkinson's disease, or is there another trigger that basically causes constipation, and then this would eventually lead to Parkinsonism? Or there's actually also experimental evidence that you can induce constipation by making a central lesion in the basal ganglia, and you can actually cause constipation also through that route. So there is not yet, I think, it's not completely established in which direction these things develop. I think there is a clear association. We're here very interested in the microbiome because I think it has become clear that the microbiome is influencing human health in immense, diverse ways. So it has about 150 times the gene content than our own genome. So basically, of the genes that we carry with us, only 1% is actually human genes. 99% is microbial genes. And so there is a huge potential for physiological interactions. And it has turned out that there's an intense interaction between the microbiome and the brain as well. So it influences.
Could you just, Philip,
yes.
just for those who are not into the gut microbiome, you just explain what's meant by that term?
Of course. So basically, it's the whole community of microbes that we carry with us, and the vast majority of these are located in the gut. So, and especially in the colon, where we have the highest concentrations of bacteria. It's not only actually bacteria; it's also viruses, protocytes, and also, for example, archaea. But the vast majority seems to be bacteria, and this is actually the kingdom that is most studied. So frequently we refer to actually the bacteria when we talk about the microbiome. There you can also make a distinction. You could say the microbiome is basically the microbes and their environment, so the combination of this. And the microbiota is just referring to the microbes themselves, so to the microorganisms. But we're frequently using this in a synonymous way. And so essentially, as I said, this microbiome, we're just beginning to understand what effects it has on our organism. And in terms of the brain, we know that there is several pathways that seem to be relevant here. So one is, for example, the immune system. So we know that the gut is the actually largest immune organ, and it's probably the first couple of years, maybe 3 years of our childhood that, for example, can determine whether we have a propendency for allergies and these kind of disorders. And that is probably related to an effect of the microbiome on the development of immunological tolerance. So in the way we can actually deal with external microbes and whether those induce some inflammatory reaction, which may predispose to actually autoimmune diseases. So the immune system is one important aspect. The vagus nerve is probably, is clearly involved. We know that bacteria directly have actually effects on nerve endings of the vagal nerve.
I was thinking we need to introduce that term a little bit. So,
yes.
So Viviane, could you just explain to everybody what's the connection between the vagus nerve, the gut, and the brain?
So the vagal nerve connects the brain to the gut, and so there are fibers coming down from the brain and also going back up to the brain. And so this nerve is one of a very, very long nerve, and it connects from the GI tract, so different parts like the stomach, the small and large intestine, the esophagus. It connects these different parts and also touches the heart as well. And it connects this to the brain, and it connects at this site called the dorsal motor nucleus. It's an area in kind of the lower part of the brain, in the brain stem. And it's actually, this is one of the sites to be affected very early on in Parkinson's disease. And so they see the aggregated protein that's associated with Parkinson's disease, a protein called alpha-synuclein. And so these aggregates sort of clump in this area where the vagal nerve connects to the brainstem in this dorsal motor nucleus, and kind of spreads from there. So it's thought that maybe the vagal nerve is important to the origins of Parkinson's disease because of its propensity to develop these aggregates very early on in disease, and maybe the aggregates are actually originating not from the brain, but maybe coming from the outside, from tissues that the vagal nerve is connected through. And the way that this protein works is a little bit like something called like a prion. It sort of moves across different nerves and can kind of travel up the vagal nerve and eventually reach the brain. So in theory, if you were to get a clumping of this protein in a tissue like the GI tract, it could travel up the vagal nerve, enter the brainstem, and kind of seed and spread from there.
So you mentioned, of course, that it innervates many organs, the vagal nerve. So I'd like listeners to think along the lines that the name vagal nerve means it's vague in its function. And why couldn't it then be that there's protein aggregation in other organs, the heart, the lungs, and so on, that are also innervated by the vagal nerve, and that the disease could start there? Is there something to say that that's impossible?
No, I mean, in all likelihood, it probably doesn't start in only one place. So in the brain, we know that one of the first two places to be hit is the dorsal motor nucleus, this connection point of the vagal nerve and the GI tract. There's another area in the brain as well as the olfactory bulb. And so Parkinson's disease is thought to be one of those diseases that doesn't necessarily have a common cause. It could start in many different locations, one of which is the gut. It could start in the nose, it could start in the heart and other regions. Right now, there's a big effort kind of being done to try to characterize where are there clump versions of this protein throughout the body. And that's still kind of very much underway. But as far as the gut is concerned, we know that many years before patients begin the motor symptom onset, there's evidence of this clump protein in the GI tract. So
let me bring in Jon in the conversation there, because how do you feel as a person affected by Parkinson's about the notion that the disease started maybe 20 years before you were diagnosed? Or even, as Philip was alluding to it, maybe you were predisposed as an infant by the microbiome that you happened to acquire?
I think it raises a number of very interesting points. The idea that one has lots of pre-symptomatic symptoms, if you will, slightly eccentric term, but it makes entire sense to me. I mean, from my own personal point of view, I suffered primarily from anxiety and depression, and one would anticipate that if one breaks it down in terms of the neuroanatomy, one would anticipate that has something to do with the inputs to the Locus Coeruleus and the Rafi complex. And
so explain to the listeners, now here comes the neuroscientist, Jon. Tell everybody who's not a neuroscientist, what's the Locus Coeruleus and Rafi?
Well, the Locus Coeruleus is the primary noradrenergic, or I guess on your part of the planet, it would be norepinephrinergic, or I think, but it's the primary noradrenergic cell body group, and it projects pretty much to the entire forebrain. It produces a diffuse projection and is involved in arousal and wakefulness. And the counter to that is the Rafi nuclei, which use serotonin as their primary neurotransmitter. And again, they project up to the forebrain, but in much more discrete areas to parts of the cortex and the hippocampus and so forth. So there are complementary connections between these two as well. And obviously, we know that drugs which block the reuptake of norepinephrine or serotonin are effective antidepressants. So the correlate of that is that depression might be the reverse of that due to lower levels of norepinephrine and serotonin in the brainstem. And of course, that would make entire sense with the knowledge, with the understanding that the Alpha-Synuclein aggregation moves essentially up through the brainstem, and that would be entirely consistent. I certainly experienced quite noticeable periods of depression before diagnosis.
And it's notable that there are now experimental trials using vagal nerve stimulation in depression, akin to what you use for Parkinson's disease, but there the electrodes are inserted into the brain.
Yes.
So definitely, yeah, you could expand on that if you want to.
No, well, I didn't particularly want to expand on it, but I think you covered it perfectly well. But I think it's interesting in the context of the vagus, just to go back a little bit. Of course, they have used vagotomy, sectioning of the vagus in the past, as treatments for things as trivial even as hiccups. And it would be very interesting to know essentially whether those patients had a lower incidence of Parkinson's. And I think the evidence, if I remember rightly, is that they do.
That's correct. So there are 3 published studies on this, and there's 2 of which that concur, that say that severing the vagal nerve, especially if you do a full truncal vagotomy, so you cut all parts of that vagal nerve, that is associated with a lower risk for Parkinson's disease. And then there's one negative study, but the other two show a significant decrease in risk in vagotomy associated with Parkinson's disease.
So this is truly fascinating that by cutting the highway that connects the gut to the brain, you actually can lower the risk for the disease. Now the question that I'd like us to drift into, I'm going to ask Filip to talk about this and then Viviane take over, is why then does the disease initially start in the gut? What's going on? If the microbiome is involved, are there things that the microbiome produce that affect alpha-synuclein? Or is it possibly a systemic inflammation? So there are things coming through the blood circulation. Or then I'd like Viviane to speak, when you've answered those questions, Filip, about the idea that perhaps things that are produced in the gut change gene expression in the nerve cells in the gut. So the nerves in the gut behave differently due perhaps to epigenetic regulation. So Filip, what can the microbiome produce, or what does it produce every single day as we sit here? And when could you say it goes wrong?
Yeah, so with regards to the microbiome, I think one of the most important findings that we had since we started, we and others started the microbiome studies, is that no one really found, let's say, a Parkinson's bug. So like a bacterium that would sit in the gut and would be the reason for Parkinson's disease. But what essentially was described now in several studies is that actually what we call commensal microbiota, so basically normal inhabitants of the human gut, are shifted in their abundance. So we see a shift of the relative amount of certain bacteria in relation to others. And so, for example, the most reproduced finding is an increase of a bacterium that's called Akkermansia and a decrease of a bacterium that's called Prevotella. And so basically in everyday medicine, these bacteria do not play a big role because they're not pathogenic, so we don't see them causing any diseases. But it has actually turned out that both of these bacteria, for example, are involved in the metabolism of the gut mucus layer. So the gut barrier, as it is also called, is basically protecting our body and tissues and blood from potentially toxic or other harmful substances and molecules that we may ingest. And so one part of this barrier is a layer of mucus that basically prevents these molecules from penetrating towards the epithelium. Another part is connections, so-called tight junctions between the epithelial cells that basically regulate how water and other molecules can diffuse through the wall of the colon towards the deeper tissues. And there is evidence, preliminary evidence, but still encouraging evidence that actually this barrier seems to be disturbed in Parkinson's disease, in the sense that there is a more unselective penetration of molecules from the gut into the bloodstream. And so basically these bacteria that I mentioned theoretically could be related to these kind of changes. There's also bacteria that have been shown to be reduced in Parkinson's that are called Faecalibacterium and Blotia. And so these bacteria are known producers of so-called short-chain fatty acids. And short-chain fatty acids, as for example butyrate or acetate, are produced by the bacteria from fiber that we eat with our diet. So basically plant-based fiber. And so we ourselves and the enzymes that we produce in our gut, we cannot digest this fiber. But the fiber basically has been transported to the colon, where then these bacteria metabolize the fiber and produce these short-chain fatty acids from it. And there is quite strong evidence, actually, although in terms of Parkinson's, it's a bit controversial, but the vast majority of studies in other contexts suggest a beneficial role for short-chain fatty acids, in the sense that there is anti-inflammatory effects of these molecules. There's actually also epigenetic effects, about Viviane probably can talk more. So they have a direct effect on the immune system. And there is also one study that has shown that these short-chain fatty acids are actually reduced in the feces of Parkinson's patients. So there is an indication of a reduced production of these short-chain fatty acids. And so I think those are probably currently the two most interesting pathways that could connect pathophysiological findings in Parkinson's and the microbiome. And so there is evidence.
So could I ask, What
Yes.
do you think happens if I drink this? And I know you're in Europe, so you'll wonder if this is a watering can for plants, but since I'm in America, this is a cup of coffee.
Yeah. Well, coffee has obviously been shown in several studies to have a protective effect against Parkinson's disease. So at least it's associated with a decreased risk, and there is some dose-response relationship there. And actually, coffee contains surprisingly a lot of fiber. So there is actually a lot of fiber in coffee, and so that is one mechanism that has been suspected that could explain it. But obviously, coffee also contains a lot of other antioxidants and so on. So we don't actually know what the mechanism is, but there's definitely.
So what happens to the microbiota if you drink a lot of coffee? Is there a profound change, or is it just subtle difference?
Yeah, there's not too many. Well, I don't think there's any human studies about it. There is animal studies that have, if I recall correctly, actually shown that bacteria such as Prevotella may even increase. And so you have a shift to a more anti-inflammatory state of the microbiome. So but it's only a few studies that have actually looked into this specifically. Interestingly, there's similar data also with regards to smoking. So tobacco smoke, and actually also it is also known that smoking is associated with a reduced risk of Parkinson's disease. And animal models have shown that smoking also influences the gut microbiome. And the changes that have been described, there is some similarity to what we have seen in Parkinson's, but it's still out in how relevant this is for the human case. But I think one intriguing hypothesis how basically microbiome changes could be related to, let's say, alpha-synuclein pathology that we see in the enteric nervous system. So in the nervous cells that basically are in the gut wall and that regulate the contractions and also secretion of the gut, is that there is description of a low-grade inflammatory state in the gut mucosa. And so
that's when I'm going to let Viviane talk about that. Yes. But before I do that, I just got a question here. Does Parkinson starts in the gut, does that theory apply to MSA? And we have to introduce what MSA is. That's multiple system atrophy. It's another movement disorder, or classified as a movement disorder, another complex disorder of the nervous system that has some similarities to Parkinson's in terms of symptomatology, but it's more rapidly progressing and has many additional symptoms, in particular in the autonomic nervous system. And the cellular pathology is a bit different. It's mostly supporting cells called oligodendrocytes that are affected initially in MSA, whereas we know in Parkinson's it's mostly nerve cells. So does this apply to MSA? Well, I know there's a paper describing very preliminary evidence in a small number of people, changes in gut microbiome in MSA. I'm sure you know about this, Philip. But one could say that in MSA, it might not be the same parts of the gut. Not the whole gut is innervated by the vagal nerve. It stops towards the distal end of the large intestine. And the anatomy of MSA pathology would actually not so much support that it is the vagal nerve, but it doesn't mean that it can't be an inflammatory response in a visceral organ. So another part, lower part of the large intestine, or other parts of the visceral organs in the urinary tract, for example. So it's a great question, and the answer is I don't think we know. But Viviane, now, Philip talked about those local inflammation changes in the gut and how that might affect synuclein. Can you explain in layman terms how the levels of synuclein can be changed epigenetically, and what is epigenetics?
Mm-hmm. So one of the compounds that Philip talked about was a short-chain fatty acid, and this is an example of an epigenetic modifier. So just to tell you a little bit what epigenetics is, at the DNA level, there's different ways of controlling gene expression. There's the DNA code itself, and so the genes that you inherit from your parents, but there's also this secondary code called the epigenetic code. And it sits on top of DNA and is sort of like a gateway to accessing the DNA. So it sort of opens and closes the door to the accessibility of DNA. And when it opens the door, readers of the DNA can come in, read it, and the gene can be expressed, and that changes the way that cells function. Now, the interesting thing about the epigenetic code is it can be inherited from your family, but it can also be changed by the environment. So an environmental signal, like a short chain fatty acid, actually rearranges some of that epigenetic code and changes the way the genes work. So when we think of epigenetic code, we're thinking of environmental factors and inherited factors. And what's really interesting is that what you're exposed to is your environment, and as you age, changes this epigenetic code, and it can change to cause increases in alpha-synuclein, which is a risk factor for Parkinson's disease. So there's evidence in the brain, in the substantia nigra, which is where the dopamine neurons die, that there's an elevation of alpha-synuclein expression, and it could be related to a change in the epigenetic code. So there's sort of a release of these DNA methylation marks, the type of epigenetic code mark. And so a release of the epigenetic code on this alpha-synuclein gene causes too much expression. And when the gene is highly, highly expressed, that's a risk factor for Parkinson's disease.
But this is not irreversible. So the epigenetic code is dynamic, right?
That's right. So it can be changed for the good or for the worse. So for things like exercise and getting lots of sleep, this changes the epigenetic code. Making a memory changes the epigenetic code. There are lots of healthy things that you do that changes the epigenetic code. On the flip side, there's also things like stress and poor nutrition and pesticides, and these things change the epigenetic code as well. So for example, if your GI tract was exposed to a lot of pesticides or was exposed to some gut bacteria that were releasing some I'm going to say metabolites, but releasing some chemicals that affected your epigenetic code, that would change the way that cells are working inside the GI tract. And that could change the levels of alpha-synuclein expression. And if there's too much alpha-synuclein expression, there could be an increased risk for Parkinson's disease, because this protein has a propensity for clumping, and we know that it can travel across neurons and potentially travel up the vagal nerve to the brain.
So I just got a question from Simon Stott. Some of you know Simon as one of the moderators on the Parkinson's Interest Research Group on Facebook, a fantastic place to read about research news. He's also an amazing blogger, has his own blog about Parkinson's disease research when he's not doing his daytime job, where he's a postdoctoral researcher, I think, in Cambridge. But Simon wrote, smoking is associated with reduced risk of PD. Could this be related to the microbiota of the gut? And I think Philip really answered that. That could be the case. And could cessation of smoking detrimentally affect the gut? And I think the answer to that question is that the protective effect of smoking, one would have to assume, is going to be decades before the person being diagnosed. And stopping smoking.
I want to mention something related to the epigenetic code in cancer. So there's a lot of stuff on smoking and cancer and the epigenome. And actually, you can see memories of smoking for 20 years out in the epigenetic code. So you could see smoking, but it would take some time for the epigenetics to become, and therefore the way that the genes work, to completely not reflect a non-smoking behavior. So the epigenetic code has a memory, and it has a memory of things like smoking. And so therefore your genes can be influenced even after you cease doing something.
That doesn't mean you shouldn't stop smoking.
Of course, yeah. but it means that if smoking is reduced, it has a reduced risk with Parkinson's disease. Even a past smoker may have a reduced risk for Parkinson's.
Okay, interesting. And we're going to go to John, who has a question.
Yes, I hope I'm not sort of derailing the conversation, but it's a question really for Philip more than anyone. It relates to what was said earlier. I grew up in the 60s, and at a time when there was, well, amongst many other interesting things, there was also constant and gross abuse of broad spectrum antibiotics. And I'm just wondering whether there's any link between the kind of use of antibiotics there and the damage they would have done to the microbiome. In terms of Parkinson's down the line, whether there's any known link. And I have a related question to that, I might as well get in when I can, whether or not there's any effect of Crohn's disease as well in Parkinson's.
That's a great question. John, just as a sideline, I was relieved that you ended the sentence constant and gross abuse of with antibiotics. When you're talking about the 60s, I thought you'd talk about something else. But
well,
Philip,
we've all been there, Patrick.
Well, not me. Philip. Oh, you're muted, Philip. I think you have to unmute yourself.
I'm sorry. Yeah, very excellent, important question. So, short answer, that, to my knowledge, for Parkinson's, there is no study that has looked into that. Here in Finland, we are in the lucky position that we have quite good registries starting at least in the 19th, certain registries also already in the 80s. So we're actually currently looking into that issue, and we hope that we will have something to publish by the end of this year about the potential connection between antibiotic use and Parkinson's disease risk. There is obviously evidence that, in particular, if in childhood there was a increased use of antibiotics, that it can affect, for example, the risk for obesity and also for certain allergies and so on. So we know that there can be long-lasting effects of an intensive antibiotic use. But in terms of Parkinson's, I don't think there's too much evidence. I think minocycline at some point has been studied even as a drug for Parkinson's disease, so it's also an antibiotic, but it has also, I think, independent anti-inflammatory effects in relation to the antibiotic effect. So that's all I know about this connection. In terms of inflammatory bowel disease, there is actually now several studies that have looked into this. The evidence is a bit conflicting in the sense that, if I recall correct, there is now 2 studies that have suggested positive association, so an increased risk of inflammatory bowel disease, increased risk of Parkinson's disease in patients with inflammatory bowel disease, but also one study that found actually a decreased risk of Parkinson's disease. One interesting aspect in those studies that found an increased risk is that that risk seemed to realize itself only in patients that did not receive so-called biologicals for their treatment, for the treatment of their inflammatory bowel symptoms. So in the sense that this may even prompt discussion about the use of such anti-inflammatory medications, maybe as a treatment for the prevention of Parkinson's disease. But
couldn't you mention the particular anti-inflammatory treatment they studied? Because it wasn't your regular off-the-shelf anti-inflammatory drug. It was a very fancy antibody.
Yeah, I think it's TNF alpha inhibitors, probably etanercept. Well, I'm not. Yeah, so in that sense, it's not just taking cortisone. So you need the evidence is more for these more advanced treatments. But the number of studies is really small at this point. So but it's obviously encouraging to see such an interaction between the treatment and the actual effect on PD risk in this population.
So, Viviane, do you think that means that anti-TNF antibodies, the therapy that Philip was referring to, could be used to treat people who already have Parkinson's? Or is the party, so to speak, over by the time you have motor symptoms? Is it too late to treat the potential inflammation in the gut?
So inflammation is thought to kind of occur early on, even before motor symptoms have begun. And so that, in order to kind of stop the loss of dopaminergic neurons, the sweet spot would be before the clinical motor symptom onset, these treatments with TNF alpha. But neuroinflammation does persist even after the onset of motor symptoms. So there could be some benefit to the remaining surviving neurons to kind of prevent what's left from further loss. But really the best window for a lot of these would be before there's this kind of massive loss of dopaminergic neurons. So keep in mind, by the time that the movement symptoms onset, you have about a 50-60% loss of these neurons. And so there's a massive loss that has already occurred. And if you're really kind of trying to block inflammation and inflammation that would eventually lead to these the cell death, it would be done years before the movement symptoms would occur. That's not to say that there is an added benefit of the 40% that are remaining, and to try to keep those alive. So I also wanted to mention, so we talked a little bit about antibiotics, but antivirals have also kind of had some evidence to show that antiviral therapy could be beneficial for neurodegenerative diseases. And so there's the bacteria, but there's also the viruses. And so preventing viral infections could be beneficial as well. And in other types of neurodegenerative disease, there's evidence that viruses can sneak into the brain and kind of have an increased abundance of these in the brain and be localized inside of or around the nucleus, around the center part of the cell of the cells, the neurons, the real workhorse of the brain. And so there's the bacterial component, but there could also be other things like viruses and fungi.
We actually had a question by somebody called Felix Nadora. What's your opinion of the research published by Dr. Roger Durmashkin suggesting that an enterovirus could be a cause of Parkinson's? And I think he's referring to a small paper in the journal F1000 where they show some electron microscopy suggesting that there is a virus combined with immunohistochemistry in autopsy specimens of Parkinson brains.
Mm-hmm.
This, of course, is a great question. Yeah, could you maybe talk a little bit about the idea that there could be a virus in the gut and the work of Sasloff and collaborators in that regard?
Right. So viruses can be in the gut, but they can also kind of travel through the bloodstream. And so some people think that there is some evidence that in Parkinson's disease, the barrier between the blood and the brain is leaky. And so these viruses that are kind of going through the blood can kind of sneak into the brain, or maybe the cells that have been activated, the immune cells that have been activated in the blood can kind of enter the brain and harm dopaminergic neurons. Now, this work that Patrick referred to shows some electron, it's like a close-up images of cells in the brain, and they see this kind of abundance of certain types of bacteria. And it sort of follows a lot of the evidence in Alzheimer's disease, where there is evidence showing that there is an increase in bacterial pieces, these peptides, that are located inside of the neurons. And there's also other viruses like herpes simplex virus, and there's an increased abundance of these in Alzheimer's disease. And in
Parkinson's, there are hints of that too.
That's right. So it seems that the vagal nerve could be involved in the passage of things like alpha-synuclein, but the bloodstream and this leakiness between the blood and the brain barrier, this could either be letting in immune cells that are active or be letting in viruses, and that could be causing harm to the brain cells as well.
So we have a couple of questions that I'm going to come to. So just those of you who've posed those questions know that I'm seeing them. Richard Windle has one, Massimo Marano. In fact, I may take Richard's question now, and this might be a way for us to begin to wrap up the gut aspect of a starting point for Parkinson's. And Richard asks, these are some very interesting hypotheses, he says. How can we take these forward? What might we do to quote unquote, prove that cells, fatty acids, the gut, the vagal nerve are really related or not related to Parkinson's? Do we have theories, or are we merely spectators? So I think Richard is asking for what sort of laboratory or clinical observations can we do to take this field forward? And Philip and Viviane and Jon, you're welcome to discuss, what will be the next crucial experiments or studies.
Yes, I think we have actually seen already first encouraging experiments in animal models that have basically encouraged us in the sense that there could be actually a causal connection between the microbiome and Parkinson's, because all human studies that we have are only cross-sectional studies. So we cannot really make any decisions, or we cannot conclude whether the microbiome changes, for example, are...
So how do you do that in a laboratory? How can you show something's causally related? Give us just an example.
Yeah, well, there is studies, for example, in a certain mouse strain, so in a transgenic mouse that is overexpressing alpha-synuclein and then develops Parkinson-like symptoms, and it actually also develops alpha-synuclein pathology and also neuroinflammation, and it's also basically having constipation. And so there is studies from Caltech, from the group of Sarkis Mismanian, that have shown that when you raise a germ-free version of this mouse model, then this model basically has less motor symptoms, less neuropathology, and less neuroinflammation. And the same was seen also when these mice were treated early on with broad-spectrum antibiotics that basically eradicated the microbiome. So essentially, this study suggested that by removing the bacteria from the gut, you get an alleviation of these Parkinson's symptoms and certain aspects of the Parkinson's pathology. And they also showed that by reintroducing either a mouse microbiome or actually microbiota from Parkinson's patients, you could basically reintroduce the pathology. So the motor symptoms would come back, and also the neuroinflammation, alpha-synuclein pathology. And so they extended this as well to short-chain fatty acids, and they also, instead of transplanting the microbiota, they simply fed these mice a certain mixture of short-chain fatty acids, and that was able to induce basically the same motor symptoms and pathological changes as the microbiota did. So their conclusion was that the short-chain fatty acids are one likely pathway that could connect microbiome changes and Parkinson's pathology. However, while this is, I think, a very important paper because it shows the possibility to influence these Parkinson's relevant processes by manipulating the microbiome, it is still quite controversial because when the vast majority of the literature suggests that short-chain fatty acids are beneficial for health. And there's also animal studies in other Parkinson's models that have actually shown that butyrate can alleviate motor symptoms in certain animal models of Parkinson's disease. So the reason why this divergent result was seen in this in the Caltech study, I think, is not clear. But I think the most important take-home message is that there is evidence for an actually causal connection in this animal model. And so there is good reason to believe that this could also be possible in human cases. So
Jon and Viviane, what would be the next experiment? I think you're both aware of this Sarkis Masmanian paper, which was, received a lot of excitement and attention about a year and a half ago when it came out. So what's the next experiment to take that further?
I think we have identified these changes in gut bacteria. Now we have to understand how they increase the risk for Parkinson's disease. What is the exact mechanism by which they do this? One example of this is to try to understand the interface between the gut bacteria and human gene regulation or the human immune system. So what about these changes in the microbiome does to cells to kind of increase the production of alpha-synuclein or cause changes in gene regulation that could eventually lead to Parkinson's disease. And then the other really important decision is piece of information is how does, for example, clumped alpha-synuclein decide to leave the gut and go to the brain? What is the kind of exact mechanism by which that happens? So, for example, it could be changes in the way that the sort of garbage disposal system of cells works. So if there's less clearance of this protein, maybe there's more of it accumulating and it can travel to the brain. Maybe there's another trigger and we haven't really found that. So I think...
So is it possible that we are constantly producing these aggregates in the gut? And when young...
That's right. So there is some evidence that shows that every time you respond to infection or responses to infection involves an upregulation and increase in alpha-synuclein. And so this would be kind of the normal version of alpha-synuclein that everyone has. And so there seems to be kind of an interplay between infections and inflammation and this protein that's associated with Parkinson's disease. Now, when does the protein decide to go from a healthy state to kind of a clump state that could eventually travel to the brain? We don't fully understand that process. Could it be that it just happens by chance, or could it be that there are other mechanisms, either genetic, epigenetic, environmental, that needs to have this kind of double hit, and it allows this protein to kind of misfold and eventually go to the brain, for example?
Could it even be that under certain circumstances, the immune system uses the misfolded variants of synuclein to defend against foreign pathogens?
I don't think that that's very well known yet, but that could be something that I know that the normal form of alpha-synuclein is a way of recruiting the immune system. So it's sort of like whenever there's a lot of alpha-synuclein in normal state, it's sort of like a calling card, and it calls over immune cells to come fight the infection. Now, whether there are different forms of alpha-synuclein, the clumped form versus the healthy form versus the cut up form, whether there's different variants of alpha-synuclein that are more effective at doing that. There's not a whole lot of evidence showing that, but I think that could be another research avenue to be explored.
There was a paper just about a week ago discussing the different chopped up forms of synuclein and how some are more prone to aggregate than others. So Jon, do you have thoughts about the next steps, you know, as a person affected with the disease, and also as if you said former neuroscientist, I think you're neuroscientist forever if you've been one once.
I think it's there are many challenges, I think, in terms of the next steps. One of the experiments that just came to my mind is if you're looking at the modification of the gut microbiome and its relation to causality of Parkinson's, it would be very interesting to know what, for instance, anti-synuclein antibodies as a treatment would do in terms of blocking that pathology. And the other thing, I just wanted to make a minor aside. We've talked, and I think the general public often conceives the brain as being entirely neurons, and I think it's just worth just reminding them that the neurons are only 1 in every 10 of the cells in the brain. There are a great many more cells, and many of them immune cells than neurons. And we kind of, they're almost the unseen handmaidens of the neuron in some ways, but their functionality, I think, is often grossly overlooked, but obviously less so in the context of synuclein pathology, I think.
So there's a paper, I think that's a very good point. There was a paper from Hopkins in July this year where they tried a variant of exenatide, the anti-diabetic agent that has shown some promise in people with Parkinson's, potentially slowing the disease a little bit. They tried a variant of this in a synuclein mouse model, and they showed that it inhibited the propagation of synuclein aggregates, this spreading, but it actually seemed to do it by affecting the support cells. Both microglia and astrocytes were altered. So you're absolutely right. We frequently overlook the importance of these abundant and very likely important cells. With regard to your mention of the synuclein immunotherapies, I think it's encouraging to know that there are several big pharma companies and a couple of small pharma companies that are already in the clinical arena testing these different types of immunotherapies. And, you know, 2 to 5 years from now, we'll be getting more and more news coming out and learning if reducing synuclein in the extracellular space is going to be a beneficial thing, and if there are certain types of synuclein we want to reduce. So we have about 6-7 minutes to go, and I'd like to switch gears a little bit. I'm scrolling through questions that have come in, but I mentioned that Massimo Marano had a question about something. He says, do you think that a disease somatotopy could start in the gut? I think he's referring to the fact that the pathology tends to follow a certain pattern in the brain. So early in the disease, we've heard several of you talk about the dorsal motor nucleus of the vagal nerve being one of the first areas affected, and then that multiple areas get involved, and the substantia nigra, where all the motor symptoms might come from, is only affected after a few years. And his question goes on to say, could alpha-synuclein, the vagus highway with respect to another vagus way? So I think really the question is there another way that synuclein pathology could start that would then affect other brain regions? And it might be a good segue over to the nose and the olfactory system. We said at the beginning of this webinar that we talk a little bit about this. And we know that if you ask Tom Veitch, who's a very prominent neuropathologist, he will say that alpha-synuclein aggregates are almost always present in the olfactory system. And in his mind, he thinks the nose or the olfactory system is the first site in most people. So we also know that people who present with Parkinson's very frequently have a loss of sense of smell, and they can frequently trace that back, at least anecdotally, for 5-10 years, that they couldn't taste the pizza that they always used to taste. And the question then is, could alpha-synuclein aggregates begin somewhere in the nose, and they could then possibly spread through the olfactory system, eventually reaching the midbrain where the motor symptoms occur. And could this also explain perhaps why some people with Parkinson's have a more aggressive course than others? Could it explain why some people may get cognitive symptoms earlier in disease, whereas others, there won't be any cognitive symptoms for over 25 years? So the starting point, be it the vagal nerve or the nose, could govern this. Does anybody have any comments on this notion? If not, I can also change the question a little bit. I mean, Philip, you know that the microbiome of the nose, the nasal microbiome, is also very complex and somehow related to the gut. So could there be changes in nasal microbiome that's a trigger?
Yeah, well, we looked at it, and another group from Luxembourg also looked at it, and unfortunately, neither of us was able to identify a change in the composition of the microbiome of the nose. But we have to admit that it is actually technically much more challenging to analyze the nose microbiome, and obviously, we also only looked at the bacterial component of the microbiome in the nose. But it's much more difficult to get samples from the nose, especially when you try to sample from close to the olfactory bulb, because what we know is that it does not make sense, for example, to sample from the anterior part, from the outer part of the nose, because it's a completely different microbiome that you have close to the nostrils in comparison to what you see deeper in the nose, closer to the olfactory bulb. So essentially, 2 studies were negative, but I don't think that it rules it out that there would be a change. It could be also technical issues that are related to that.
Could it also be because you presumably sampled the people who were aged 60 and had Parkinson's for 5 years? Perhaps you wanted to sample the nose of those people when they were aged 35. And is it possible that they had a change in microbiome that lasted for 1 or 2 months related to some infection, and that triggered the synuclein aggregation that then took 5, 10, 20 years before it was propagated into the brain?
I think it's definitely possible. I also think it's extremely difficult to study. You would need huge cohorts that you need to follow up for a long time to do these kind of studies, and you would need to do repetitive sampling to catch these temporal changes. But it is definitely possible. We know that also antibiotics disrupt the microbiome for a certain period of time. Most patients will return to their previous state of the microbiome within 1 to 3 months. But we also know that there is a certain small fraction of patients that will actually never return. They will never get their old microbiome back. So they lose certain parts of the microbiome forever. And so these are all things that may be very relevant, but we just don't really know if they have a role in Parkinson's disease.
So we're entering almost the last minute. So I promised Viviane, I'll give you 20 seconds because Jon wanted 20 seconds too. So what do you think about the nose? Do you think it's a reasonable hypothesis that it also could start there?
Well, just to kind of build on what you were saying, so there is evidence that transient trigger-like events can occur. For example, traumatic brain injuries. These are things that are reoccurring. That's associated with an increased risk of Parkinson's disease. Pesticides, which is something that you're off and on exposed to, are also associated with an increase in Parkinson's disease. So it's very possible that, say, if you were to inhale a lot of pesticides, that could be associated with a misfolding of this protein. And so there is evidence that if you inject misfolded protein into the olfactory bulb, that it can travel retrograde. It can go through the brain and then cause more pathology. And I think there was someone named Patrick Brundin that might have shown evidence for that. But yeah, so in animal models, you can see this kind of ability of misfolded alpha-synuclein clumps, these clumps associated with Lewy bodies and the pathology associated with Parkinson's disease, and it can travel from the brain, the part of the brain that innervates the nose, and go back and eventually at later stages reach the substantia nigra.
So you got more than 20 seconds just because you mentioned that study, so thank you. So, Jon, you have 20 seconds.
Has anyone looked at the effects, the prevalence of Parkinson's in people who have been exposed to long-term nasal irritants and chemical industry, for instance, that kind of thing. I'm thinking also people who work in morgues are exposed, of course, to formaldehyde, which is a notorious nasal irritant. And I wonder whether any of the things like that, which cause degree of edema in the within the nasal epithelium, whether that is a reasonable vein to look at, as it were, in terms of also different types of Parkinson's as well, obviously.
So epidemiology, as you know, is notoriously difficult because you need very large sample numbers and there has to be an accurate history. I'm not personally aware of studies on formaldehyde and PD or Parkinson's. Philip and Viviane, do you know?
Well, no doubt inflammation is associated with Parkinson's disease. So if you have something that's, an irritant and is causing chronic inflammation in your nasal passages, that would be, could be a risk factor, but there's very little evidence of that so far. But there is knowledge that activation, unusually high activation of the immune system is associated with Parkinson's disease.
So I've been told we have to sign off, and before we do that, I'll just say a couple of topics we didn't cover that I think we can cover at a later webinar, but that's the notion that the shape of the misfolded synuclein can also influence the type of disease, so be it multiple system atrophy versus Parkinson's, or Parkinson's with dementia versus Parkinson's without dementia. And we can maybe talk about that at a later date. And also another notion we didn't really touch upon so much, but the bacteria themselves can produce misfolded proteins that have recently been suggested to affect the folding also of alpha-synuclein. So there could be a direct interaction there. We had a couple of other questions that came in, and I'm sorry we couldn't answer them online or live, I should say. We can perhaps type in answers to you afterwards. But I'd like everybody to say thank you to Philip and Jon and Viviane for being very excellent panelists and very informative. And also I'd like to thank those of you who joined this webinar. We will be returning, so keep an eye on the Facebook and web pages and you'll find out when the next webinar will be. So thank you to everyone for participating.
Thank you.
Bye-bye.
Thank you.
Bye.